Hello there guys, and welcome to our next class for the third unit. This is teacher Krishna, and this is a class that is part of the School 9th BYS. Just like you know, our subject is biology, and today we are working on week number 26. So be welcomed, and let's go over our presentation. Here you have the end in mind for today. Students will be able to review what they have learned in unit number 5. Basically, the activity that you need to complete today is to finish the review and assessment from unit number 5 in order to complete a module assessment that is going to be online. Here you have a small summary of the activities to complete. Complete the review and assessment. You will find it in pages 157 to 159. Study the questions that you find in there. Complete the quizzes game during the live session. So right now I'm going to help you out with a half of the questions that you find in the review and assessment, okay? And you have to complete the other half. And during the meet session that we have today, I'm going to share with you a link as well as a code to access an online quiz that you need to complete. And based on your results, I'm going to assign you a grade today. But it is always important that you can upload a picture of your results to Educativa, okay? So make sure to have your book right now because we're going to start completing some of the questions. And you can notice that it is something different, right? Because actually I took the pictures with my cell phone, so you can actually notice that the images are a little bit different, but don't worry, we are able to read the results, and of course we can help you out in there. So question number 1 from lesson number 1. Which human trait is controlled by a single gene with multiple alleles? And based on what we have studied in previous topics, you will notice that it is blood type. Of course, we are going to have the recombination of many different alleles from the father and from the mother, and at the end we are going to generate a special blood type. Number 2, color blindness is carried on the X chromosome and is more common in males than in females because it is a sex-linked trait. And remembering a little bit about the structure of the chromosomes, remember that the X chromosome is going to have way more information than the Y chromosome because it is larger, basically. And since the information from the X chromosome is going to be more substantial in the new organism, it is way more possible that if it contains a color blindness section, then this type of allele or this type of trait is going to appear in the new offspring. Let's go with question number 3. Compare and contrast. Describe the main differences between the inheritance patterns for a dimplet gene and for height. So this explanation is going to be a little bit long, but here you have it. So a dimple gene is the result of a single gene with 2 alleles. And a person either has dimple gene or does not. In contrast, height is the result of multiple genes, and so there are many different heights that are possible. Just like we have studied before, when we study a certain trait, what's going to happen is that some of them are going to be defined only by a pair of alleles, but in some other occasions, multiple sets of alleles are going to be part of the production of this new trait in the offspring. Now let's go with question number 4. Interpret data. Complete the Punnett square below to show the possible genotypes for the offspring of a colorblind mother and a father with normal vision. Circle the genotype that will produce colorblind offspring. Okay. So remember that since colorblindness is a sex-linked trait, we need to analyze the chromosomes that are specifically related to the sex of both the people that we're analyzing. When we talk about male, remember that we are going to have a combination of X and Y. Just like you can see right over here, this will be the representation of the sex chromosomes of a male, X and Y. If we go back to the chromosomes that are, of course, related to the mother, we're going to have a X and X. All right. And whenever we analyze that a specific trait is carrying what we are tracking, that in this case is color blindness, we're going to add a letter C that is going to be representing the carrying, let's say. Okay. So in this case, in the first one, we're going to have a recombination of X and X on the first square that we have here. On the second one, we're going to have X and Y. In the third one, X and X. And in the last one, we have X and Y. And just like we have studied before, color blindness is more typical from male. So in this case, the two babies that are going to have a color blindness are both going to be boys. And so we have to circle them right here. Okay, that will be for question number 4. Let's go over the next one, question number 5. Which of the following will most likely be used to diagnose Down syndrome? And in this case, it would be a karyotype. Because in karyotypes, we can take a look at the genetic information of a person, of course, and of course, track down the chromosome that has been changed inside the person. Then we have question number 6. Cystic fibrosis and hemophilia are 2 examples of genetic disorders. Genetic disorders are modifications or changes in the DNA of a person. Then number 7, make generalizations. What information is shown by the karyotype? Basically, the number of chromosomes in a person's cells. And going back to the example that I was giving you about the Down syndrome, remember that the Down syndrome is caused when there is an extra pair of the chromosome 21. So instead of just being 2, we're going to have a group of 3 in chromosome 21, which at the end is going to generate an intelligence disability in the person with this syndrome. Then we have number 8, relate cause and effect. How does the cause of cystic fibrosis differ from the cause of Down syndrome? So cystic fibrosis is carried by a recessive allele that is the result of a mutation. And Down syndrome occurs when there is an extra copy of an entire chromosome. All right. Let's go with number 9, interpret diagrams. The pedigree chart below shows the inheritance of sickle cell disease. Remember that sickle cell disease is like a change in the blood cells, in which some of them are going to be a little bit larger and they can cause obstruction in the vessels of people. Circle all the individuals on the chart who have the disease. Draw a square around individuals who are carriers. Alright, so important to remember. Circle the individuals that actually have the disease. And the ones that are only carry the disease but they don't have it, we have to use a square. So we have to analyze, of course, what we have right over here. So in this case, if the person is represented by a circle or a square with only half of the inside painted, then the person is just the carrier of the sickle cell disease. So let's try to track the ones that are shown in this diagram. Right, so this one, this one, this one, this one, and this one are only carrying the disease. Okay, they are not fully painted. And on the other way around, we have a circle or squares that are fully painted, which is the representation of a person with sickle cell disease. And which ones here are fully painted, in this case, are going to be these 2 people that are right here. So you can use circles in order to represent that. Okay. Now let's go back to the last section right over here. Then we have number 10, an organism that has the same genes as the organism that produced. It is called a clone. So basically an identical being that has been produced based on another being will be just a clone. Number 11, inbreeding and hybridization are 2 different types of selective breeding. Okay, and lastly we have number 12, write about it. Suppose that you are giving a presentation about genetic engineering to a group of people who are not familiar with the topic. Write a short speech that includes a definition of genetic engineering, a description of how it is used, and an explanation of some of the concerns about its use. So, let's put it into very simple words because just like it is specified here, we are giving the speech to people that actually don't have that many knowledge regarding this topic. So first of all, we need to include a small definition about genetic engineering. So basically, genetic engineering is the scientific study of genetical makeup or genetical information to do many things. We can use the genetical engineering to, for example, improve the feeding of animals. We can improve some plants and, of course, make them better for humans. We can use genetic engineering in order to clone certain animals as well. And lastly, I will say that we can use genetic engineering in order to track certain disease. For example, the Down syndrome, or we can talk about the sickle cells as well. That'll be the definition. Oh, and of course we have a description and how it is used. Another way in which we can use it can be in forensic science to identify the DNA of possible suspects in a crime scene. And what are some of the concerns about its use? Well, a lot of people actually criticize it because there is a huge debate if it is actually correct or not to create a clone from an animal, because we have to remember that that is something artificial. In the natural world, of course, there are many babies, many puppies, and of course new offspring from several animals that are just generated naturally. But of course, when we take into account that a clone actually doesn't have the requirements to be considered natural, and that is just created by humans, some people might think that actually that is going against the nature, and at some point, if you want to see it from that perspective as well, is going against God at some point. But that'll be about it, basically. I already helped you out with the first 12 questions, guys. Remember to finish the other half of questions from your book and study them to complete the quiz during the live session. That's it for me today, and thank you a lot for coming. I will be seeing you in a couple of minutes. Goodbye, and always take care.